So there's a number of reasons why an institution might start a captive breeding program. One might be just general education purposes, for example, displays for children. Another might be for biomedical research. And finally, there could be captive programs to aid in the recovery of endangered species. And in these types of programs, the typical goal is to limit the changes that happen in those captive populations by retaining at least 90% of the genetic diversity. Because we're usually talking about an endangered species, the original populations are usually small and declining. And then once they're moved into captivity, they're held at small sizes just for logistical reasons. This means that inbreeding and drift become a really strong force in these populations, and this leads to reduced disease resistance and reduced ability for these populations to adapt to novel situations. When you're thinking about taking an animal from this captive kind of sterile environment and putting it into a wild and changing environment, then these become critical concerns for those individuals. And the other thing that happens when these populations are held in captivity is that they actually start to adapt to the captive environment. Traits that increase fitness in captivity can be thought of as a good thing, right? Your animals are doing better overall, but usually those traits are maladapted once the individuals are released. And I think the best example of this are the startled antelope. So in the wild, when they're startled, they'll sprint, and at least some of them might avoid predation that way. However, when you put some of these antelope in captivity and startle them, then they run into enclosures, and this can lead to injury or death, which is not good. So that behavior tends to decline in the captive populations. And then if you think about releasing some of those individuals from these captive populations back out into the wild, now they're startled and they don't run away and they become dinner. So what I've been working on for the bulk of my dissertation is looking at how breeding protocol, which is really how we select which individuals will breed in a population, how this influences the rate at which these captive populations change. And I'll focus today on how they mitigate inbreeding, drift, and selection. And then I'll follow up with how we can use genotypic data in order to kind of enhance some of the protocols that we already know work better than others. So in order to look at captive programs or captive populations, we first needed to get some. So we started 6 captive populations using 20 white-footed mice trapped in Illinois, and bred these populations according to one of 3 different protocols. The first was the random mating protocol. In this case, individuals were selected without respect to their genotype or their phenotype, although we did avoid close sib matings. The second protocol is the minimizing mean kinship protocol. And this one works by first calculating all of the pairwise kinship values, and that's shown kind of in the top table there, and then calculating the mean, in this case over the column, so the mean kinship for each individual, and then finding the individual with the worst mean kinship score, so the individual that is most like the rest of the population, and removing that individual from the kind of pool of breeders that are available. And then we would recalculate kinship and do it all over again until all of the individuals have been removed. And then the last 10 pairs, which would be the last 10 pairs of individuals with the best kinship values, would then be used to propagate the population. And then finally, we have this docility protocol. And this was meant to mimic the selection that occurs for easy-to-handle animals. So we selected against 2 kind of stereotypic mouse behaviors. One is this gnawing on the cage bars, which is shown in the top video, and the other one is this cutesy little flipping behavior. So we selected against these behaviors by counting the seconds that they spent flipping during a certain period and then selecting the animals with the lowest scores. So just a quick overview of how this worked then. All 6 populations were started from the same 10 wild-caught animals. And then their offspring were used to start each of the 6 populations, which were bred according to one of the 3 protocols, 2 replicates per protocol, all the way through to generation 19, where none of the lines were crossed. So the first thing we looked at was how these protocols differed in terms of accumulation of inbreeding. And to do that, of course, I need data. So I used the inbreeding coefficient calculated from the pedigree, which I could calculate over all generations for each of the 6 populations. And so before I tell you about that, I'll just show you briefly how most of my figures look. Generations on the x-axis, and then whatever I've measured is on the y. Mean kinship is shown in green, random in blue, docility in red. And then the replicate lines are shown with replicate 1 being the solid line and replicate 2 being the dotted line. So in this case, for inbreeding clearly increased for all of our populations, which you would expect for a closed population of that size. It increased the most in the docility 2 line, which actually went extinct after generation 9 due to reproductive failure. But what we did find was that the mean kinship lines, again the green ones, increased the least, so they were accumulated the least amount of inbreeding over the course of 19 generations. So from this, we can conclude that, yeah, it does seem that mean kinship protocol does mitigate inbreeding pretty well. So next I wanted to look at the effects of drift, so I needed some more data. I used a panel of 11 microsatellite loci, from which I calculated the average heterozygosity and the allelic richness on average per population and generation. And I did this at generation 0, which is really the 10 wild-caught founding mice, 6 and 19 as well, and some samples from the contemporary source population, which we trapped in 2012. So looking first at heterozygosity, what I was a little bit confused about initially was that all these values are operating like within a really small range. Even the difference between the captive populations and the wild population is not very big. At least for the randomly mating population, though, we can calculate how much heterozygosity we would expect to retain over the course of these 19 generations. And assuming an effective population size of 20, which is the number of breeders we used every generation, we expect that it should retain 78% of the diversity, and we found that on average the random populations retained 80% of the heterozygosity. So that seems to make sense. Then switching to the average number of alleles per locus, we found that on average the founders had the same number of alleles per locus as the wild population, which is a good sign again. It seems that we've captured the diversity in the wild. And we found that mean kinship kind of retained more alleles than the other populations, although not significantly so for many of the comparisons. In general, all of the populations lost diversity, and 90% of the diversity was not retained for any of our populations or protocols. So from this, we've concluded that drift still seems to be a significant source for loss of genetic diversity over time. So that kind of covers inbreeding and drift, but we're still left with, what about selection? So for this, we needed even more data. I used a panel of SNPs, which were generated using the L-Shire genotyping by sequencing method. And this resulted in 705 singleton SNPs and 531 putatively linked putative linkage groups. And from these, I've estimated heterozygosity by first calculating it for the singleton SNPs and then calculating it for a randomly drawn SNP from each of the putative linkage groups averaged over 100 replicates. And I calculated this for generations 1, which should be representative of the founders, 6, 12, and 19. In order to look for selection, I used a simulation approach. So I started all the simulations with the genotypes from generation 1, and then determined reproductive success, which is really the probability of mating and the number of offspring that each pair would produce from the empirical data. And then I mated the individuals according to the 3 different protocols. And I ran the simulation 100 times for each of the protocols. And this resulted in a allele frequency distribution to which I could compare our observed allele frequency for each of the SNPs that we had. So I calculated the p-value and then corrected it using the Benjamini-Hochberg method. And this resulted in actually quite a few SNPs that I was able to detect selection on. I used an ANOVA then to compare the number of SNPs between protocols and generation and found that really there was no difference in the number of SNPs under selection between the protocols. A little bit surprising perhaps, but what I was really interested in is, what is the effect of selection on these SNPs? So to look at that, I compared the individual heterozygosity calculated from only the neutral SNPs to the individual heterozygosity calculated from only the SNPs under selection. And what I found was that for every individual, the SNPs under selection had a smaller heterozygosity value compared to the neutral SNPs, which makes sense if most of these are a selective sweep kind of a situation. By looking at the confidence interval around the means within the protocols, I found that the mean kinship had significantly less difference between the two sets of SNPs compared to the random mating, which indicates that selection had a weaker effect on the heterozygosity in the mean kinship compared to the heterozygosity in the randomly mating. So from this, it seems that mean kinship does somewhat mitigate the effects of selection. What didn't quite make sense maybe is the docility group. We know that that population is under a strong selective pressure because we're doing it. So I might have expected it to have an even bigger difference than random. But what I think of going on is that there's only a few SNPs that were actually under selection in the docility selection protocol. So using a genome-wide association, I found that there's only 18 SNPs that are under selection and associated with the behavior. So I suspect that the strong selection on the very few number of loci within our docility selection protocol swamped the effect of the relatively weak selection across many SNPs in the genome that we saw in random mating. So kind of in conclusion of this first part, inbreeding and selection are pretty well mitigated by the mean kinship protocol, but drift is still a significant force that removes genetic diversity in all of the protocols. So for the second portion, I wanted to know if I could use some of the genotypic data that we had in order to better mitigate the effects of drift. And I did this within the mean kinship protocol, because that was our best protocol. And this is really based on the fact that pedigrees assign siblings an equal kinship value, even though if you look at a locus within several individuals or several siblings, they may or may not be identical, probably not identical. So I used simulations to estimate the impact of including genotypic data to try and tell the difference between these individuals. So how this worked was I first, just like before, calculated the mean kinship for individuals, all individuals, found the individual with the worst mean kinship score, and if there was a tie there, then calculated relatedness using our genotypic data and removed the worst individual, or the one that was related to the other individuals at the highest degree. And here mean kinship again is shown in green, and this enhanced method is in gray. And what I found was that on average, by generation 19, this enhanced protocol retained about 2 additional alleles per locus, which I think is pretty good. So it seems that making use of the genotypic data can help us mitigate drift better. So finally, selecting breeders according to mean kinship retains the maximum genetic diversity compared to docility and random mating by mitigating the effects of inbreeding and reducing the effects of selection. But we can do better by trying to identify uniqueness among individuals that may have been assigned identical kinship scores. And with that, I'd like to thank all of my funding sources, my committee, my advisor, Andrew DeWoody, who is looking for graduate students and says you should email him, as well as my lab. And I'll be happy to take any questions.